I think this one flicks it through to the next slide. But yes, hi. We're going to be talking about chromatography and the tangential flow filtration stuff, right? So, hey, it works. Cool. The pictures are super fucking fuzzy. That is hilarious. Anyway, so column chromatography is very fun. Industrial scales use like these ones, and these are honestly small for certain types of industry, but these are the models that I actually use regularly at my job. But yeah, so basically this is just most of what you need. Of course, you also need some way to put liquid in and take liquid out, but this is just most of the equipment you need to do chromatography. But we'll get there in a second. Just wanted to give you a picture of columns there. So how do they work? Well, to put it very simply, to separate molecules by some property, you need to have them interact with something. That substrate or whatever that they interact with is most often called a resin. The resins can do various different things, and importantly, there's always going to be some way for some sort of liquid or solvent or whatever to flow through or around the resin. So, the things happen in the resin. I don't remember making the slide, but the things happen with the resin. So basically, the molecules interact in some sort of way. That's what happens inside the column. So you can explain a little bit more of that. Some simple terms that I might use are up here. The elution is just the stuff that comes out of the column. The eluent is just the liquid or buffer, solvent, whatever that makes your stuff come out of the column. The column volume is just the amount of resin in the column. That can vary, and it depends on how well it's packed, but basically it's just column volume there. Polarity, if you don't know what like polar molecules are, basically, let me grab the pen. Basically, like your average water molecule, right? It has an oxygen, and it's got two little hydrogens that are attached to it. And because oxygen has a much bigger nucleus than hydrogen, it's hogging a lot of the electrons in this bond. So the oxygen is very slightly negatively charged, and the hydrogens are very slightly positively charged, right? So it actually has a magnetic dipole moment along this axis. So this side of it, the molecule is very slightly negative. This side is very slightly positive. That's basically what polarity is. The whole molecule kind of acts like a magnet. Okay. And adsorption is just when something sticks to a surface, right? You know, like absorbs when like something consumes something else, or like fully like encapsulates or surrounds it. Adsorption is, oh, I have this bead here, and there's this molecule here that just gets stuck to the surface. That's adsorption. So there's three main types of chromatography. They're split. The terminology is split on basically how they function, right? There's size exclusion, ion exchange, and affinity chromatography. And I will talk about them in that order, because it's also the order of how simple they are to how much complex it can get. So size exclusion chromatography is sometimes called gel filtration chromatography. The resin is some sort of gel. The gel is porous. And because the gel is porous, that means that like each of the little beads that make up the gel have some sort of channels through them, right? So I drew earlier an example of one that's like an ideal resin, kind of like a circular bead, but it's got like this channel through the middle, where smaller stuff can fit through the channel, but larger stuff can't. So what actually happens with size exclusion chromatography is the larger stuff just flows around the beads, whereas the smaller stuff has to flow through the beads, or it will get stuck in the beads within various interactions and stuff. And so smaller molecules move slower, larger molecules move faster. So the larger molecules come out first. They will always come out of the column first. Basically what that is, right? So to use the proper terminology, right, like they will elute first. So I can just draw a little diagram here. You'd have like a mixture, large, maybe I should grab a different pen color. You'll have a mixture of large and small molecules, right? And then what you'll end up finding out is over time, make a little graph for you. Third color. What you find is the amount of stuff that comes out over time. Kind of looks like that. Not a perfect diagram, but basically the larger stuff will exit the column first, and then the smaller stuff will exit later, because the smaller stuff will get caught up in the resin. So, I think I have another sort of a diagram, the next page, but I could talk a little bit more about it. There we go. So as you can see, it's kind of like how I just described, where they show a mixture of different size molecules. They kind of show what it does when it separates through the column. And then those show you like some type of the gel particles that the molecules might be able to fit into, right? So that's kind of what they often look like, because, you know, we can't really like precisely control exactly their shape, but they basically just have cracks and channels through those gel particles there. And you can see that the large stuff will come out first, then this middle-sized stuff, and then the very smallest stuff. Again, this has to do basically entirely with path length, right? If there's more potential paths to take, then it will take longer to take more of those paths. Because the larger stuff only has one path to take, which is around all the gel beads. So, next up is a simple explanation of ion exchange chromatography here. This separates the molecules by either charge or polarity. So you can have positively charged ions stick to negatively charged resins. You can have negatively charged ions stick to positively charged resins. Or you can have polar ions stick to polar resins, and then like non-polar ions stick to, or non-polar, they're not ions, they're non-polar. So you can have non-polar materials that stick to non-polar resins. So these charged ions, or the polar molecules or whatever, adsorb to the surface of those beads that have different chemical charges or different polarities, and then you can use a various series of eluents to get them off of the column. So what you want will stick, and what you don't want will come off. So, there. Right, so this one basically shows you one type, which is the ion exchange. So you have your different charged particles. This one's showing a positively charged resin. Right, so all the negatively charged ions that you want are going to stick to the column, and all the other ones are just going to flow through, because, you know, positive ions aren't going to be really attracted to other positive ions. And then you can add a series of either like increasing like a gradient, or just different types of eluents to try and get out of the column. So if you could read the text on here, which the thing made very fuzzy, basically what they do is they add increasingly concentrated sodium chloride to it, which can help knock the negative ions off of the resin. Because oftentimes they'll find, oh hey look, there's this very strongly positive sodium ion over here. I want to go be with it instead of be with the resin. Personifying the molecules makes things a little bit more understandable. So that's kind of what happens with here. There's also, you can do the same thing with polarity, where you can have a very polar ion like water stick to a very polar resin, and then what you can do is you can change the polarity of your solvent. Water was kind of a bad example, but basically you will elute. What's another VTEX? What's another good old polar molecule that maybe you could solve? Ethanol. Let's use ethanol. Ethanol is pretty polar, right?
Cheers.
It's got a... Huh?
Everybody.
Yeah, ethanol is pretty polar. Ethanol's got these carbons, and it's got an oxygen and hydrogen. Right? So you usually find these dipole moments in molecules kind of wherever you got these like very strong ions. So like this oxygen is very slightly negative, and then this hydrogen is very slightly positive because the oxygen wants to steal all the electrons because it's got way more protons in its nucleus than any of these other guys around here. So this is a polar molecule. That's why it dissolves in water, right? So let's say we load this onto our column. Our column is a polar column. So this will stick to the polar column. And then we start using a completely non-polar solvent like hexane. Hexane has no polarity. The entire molecule is made out of carbons and hydrogens. It is basically as non-polar as you can get. This stuff will stay on the resin because it wants to be with the other polar molecules on the resin. It will come off of the resin when you change the polarity of the solvent. So let's say you switch the solvent from using hexane to using water. Great. Now this will completely dissolve in the water and it will detach from the resin as it will elute off of the column. So same kind of deal. It's also still kind of an ion exchange because you're dealing with charges. So I did have a slide for it. Reverse phase liquid chromatography is kind of the most common thing that VTEX will do. But basically exactly like that. It will separate out your molecules by their polarity. You can have even more polar molecules than ethanol. And you can have even less polar molecules than ethanol. Less polar molecule than ethanol might be like some sort of ketone or something where you got like a carbon with 3 hydrogens, another carbon double bonded to an oxygen, another carbon with 3 hydrogens. As you can see, oxygen doesn't really have the ability here to steal a little bit of extra time on those electrons from the hydrogen because it's just bonded to the carbon. So it's even very slightly less negative. And this carbon is also very slightly positive, but it's kind of harder to get to because unlike being at the end of the molecule, it's kind of in the middle. So it's a little bit harder for other polar solvents to dissolve this guy. So that's just another example of switching it from polarity. So, affinity chromatography. This is the most fun one because affinity chromatography is just how do you separate a molecule based off of some other property that isn't its ionic charge, its size, or its polarity. So basically this is separation of molecules based off of special interactions. Some of these special interactions tend to be more biochemical in nature, like sugars binding to other sugars, certain amino acids binding to antibodies, certain proteins binding to antibodies, or certain amino acids binding to certain, like, it's hard to explain, but like the amino acid histidine just likes to bond to nickel, just nickel ions. So you could load up a column with a bunch of like nickel ion resins, and then just histidine will stick to it. And that's kind of just a special interaction, right? So there are resins can be made of a bunch of different materials based off of like whatever you're attempting to separate it for. And the substances will bind differently depending on their affinities. I put an example in for a thing that I actually do at work. We try and extract some glycoproteins from human serum. So how do you go about that? Well, use a sugar-based resin. The resin that we use is actually primarily composed of the sugar saccharose, which is not really a... You may not have heard of saccharose because our bodies don't digest that sugar, but it exists in nature, so it's helpful, right? So the glycoproteins will stick to that. But we use a little trick, you see, because there's this substance called urea. You may have heard of it. Our urine it's named after our urine because that's where it comes from. We get rid of it as a waste product in our body. However, urea really fucks with proteins. And so what can happen here with our glycoproteins or our sugar-coated proteins is that it's binded to this other sugar, but then a very polar molecule like urea comes by, and it's like, ooh, I want to be over here. There's also a little bit more complicated to it because urea can also change the shape of proteins. You may have heard that proteins can fold into certain shapes. So the urea may unfold the protein a little bit, and that also makes the interactions a little bit weaker. But basically, we can load up the column with a shit ton of urea, 4 molar, which is extremely concentrated, and then the proteins just kind of don't want to hold on to the column anymore, or hold on to the resin in the column anymore. And yeah, it's kind of how they come out. Also, like I mentioned, histidine tag proteins, their special interaction with nickel. It's just one of those things. This diagram is super fuzzy. But yeah, basically they just show that this uses some biology terminology about ligands and stuff. But basically this just shows that you can have a mixture of a bunch of different proteins, and only the ones you care about will bind to the resins, and then you can elute them off of the resins, so you can get exactly what you were looking for. So, my next part's about tangential flow filtration. This is important because of my aforementioned about the bioprocess about extracting these glycoproteins from blood serum, or human serum. Well, we use 4 molar urea, right? Our bodies do not contain 4 molar urea, right? For example, our blood is mostly just saline and blood cells, right? So we just have to recreate the saline. So how do we get rid of 4 molar urea? Well, I'll get there. We need a little bit more terminology first about this tangential flow filtration stuff. The permeate is just stuff that goes through these filter membranes. So these are not like our columns, where our columns, they're long, and then they're packed with this resin in the middle. Like, here's the resin, and then the liquid flows through. Right? It's not like our columns. So these tangential flow filters are often their own filter membranes that don't work like this. And that's going to be important, right? So we have the permeate stuff that gets pushed through the membrane. You could think of it just like a normal filter. A lot of water filters are some sort of flow membranes. Just important to distinguish from the retentate, which is stuff that's retained by that filter. A tangent line, I hope everybody here has been through at least the first level of college calculus, which was probably required, and you know what a tangent line is, right? So you can imagine that with tangential flow filtration. Did I have a diagram on the next slide? No. Okay. So you can imagine with tangential flow filtration, you have your filter here. The membrane has little holes in it for stuff to get pushed through, right? So you have your liquid flowing through this way. And because this is the other side of the filter membrane over here, you can have some stuff come through this filter membrane And because of pressure gradients and stuff, you can end up having whatever comes through flow tangentially to the filter membrane, which is the little bit I drew here in the middle. Right. So a normal like filter that you might think of just uses pressure to push shit through. Sorry for the language, but it just uses pressure to push the stuff through like a membrane, right? So anything that you don't want that's too big to fit through the filter kind of just gets stuck on the filter. But with this one, because the flows are tangent to the filter, anything that wants to get stuck to the filter membrane can actually get washed off and continue to flow through the system or exit the system. So that's kind of why it's important and different from normal filtration. So I preemptively advanced the slide here, but basically, how does it work? The filter membrane is made of a bunch of fibers. Those fibers are porous, have different pore sizes depending on what you engineered them for, what you want. Small molecules that you don't care about are going to fit through the filter membrane, or through the pores in the filter membrane, and the large stuff is actually kept in the membrane and then eventually exit out because of the nature, the whole washing it off the side. There's basically, I spelled it right. Okay, yeah. So solvents capable of being retained because of the flow rates and the pressure gradient, right? Like you might take a look at the drawing I had just drawn and be like, well, why doesn't just all the liquid go through? Well, you need pressure to push it across the membrane, and it will follow. It kind of follows the path of least resistance, right? So if there could be, if it's easier for your liquid to not go through the membrane, then it will just keep going. Some of it will still go through because, again, as with physics, all possible paths are taken. So, and that's kind of just the simple how it works. I have a little diagram here about just like the flow path. Right, this is actually notably the same one that I showed you on the first slide, where you have this holder which just deals with all the plumbing for it. You have the filter membrane here in the middle that's kind of sandwiched in between. And so what happens is you just have some sort of pump that takes one liquid from a tank out the tank through the membrane. Stuff that permeates through the membrane comes out into permeate, which will collect in a different tank. You'll have up here just a little valve that you can put back pressure on the system, because again, you're going to have less stuff flow through that permeate if you don't really have if it's not really favorable, right? If there's almost no resistance to come out the filter through the retentate, then most of the stuff you put in will just come out through the retentate. So you do have to force it a little bit. And then oftentimes you'll find that this retentate just goes back into the source container or the tank where you took the fluid from. Don't worry about that other part where it says diafiltrate, I'll get there. And this is just another small thing that's showing you about the flow is kind of like tangent to the surface. So all of the stuff that would get stuck to the surface in a traditional filter actually would just get pushed off. So that's kind of how it works. Similar kind of setup, right? You can see the pump, you can see the lines. Excuse me. But yeah, that's basically just simply how it works. I can draw another diagram after the presentation if anyone's still a little confused or wants to know a little bit more. I did that click through too many times. Hold on.
Wow.
There we go. Sorry, I just spent all that time setting it up. So what's it used for? Well, primarily in industry, it's used for dialysis. You may have heard of dialysis. You know that it's that thing that your kidneys do for you, right? It cleans your blood. It takes the stuff out of your blood that you don't want, right? Well, that is basically one way of dialysis, right? Our kidneys do it through biomechanical mechanisms rather than through, filter membranes and stuff, but similar kind of principles apply. The dialysis, strictly speaking, though, in the sense that I will be using it today, is just the replacement of one buffer with a different buffer, or a replacement of one solution with a different solution. So, like, for example, my 4 molar urea. I don't want 4 molar urea. I want saline, like the same concentration of salt ions as in our blood serum, right? That's what I want. I don't want 4 molar urea. So you can remove the stuff that you don't want through this method, and then the stuff you want to keep is, of course, larger than the pore size, right? So we want to keep our proteins, but we don't want to keep the urea. So you choose a pore size. These proteins are way larger than any normal ions are because the proteins are huge molecules, right? So you can just choose a pretty decent size pore size so that you don't have to have too much pressure. But basically, your proteins are all kept in the filter, and then all the stuff you don't want permeates through the filter. And basically, when you're doing this entire process, you can just start replacing the liquid that you're in, right? That diafiltrate I mentioned is basically just another tank. So you have your tank of saline. This is going to be backwards if I do it backwards. Yeah, your tank of saline. You have your tank urea, and then you have the who clicked it. I haven't gotten to that part yet. Right, and then you have your magical do everything box that does it for you. So basically, you just take out of here, you go into the TFF membranes, and then the retentate's brought back, and then the permeate exits out into another container, right? But while you're doing this process, you can also introduce your saline into the urea tank, and you'll have several times more volume of the saline that you want to the actual stuff that you're filtering, so that you can continuously replace as it goes. And basically just that. Then all of the urea eventually ends up over here. And over here you end up with almost no urea. That's kind of basically how the process will go, and I'll go through it a little bit more detailed in the next diagram. And then also reverse osmosis. You may have heard of it. A bunch of water treatment facilities use it. That's also one method of removing the salt from seawater to make that into drinkable water. It's a little bit different with reverse osmosis because it also uses what's called a semi-permeable membrane. You may have also heard of those. All of our cell membranes in our bodies are semi-permeable membranes. They basically selectively let across specific ions or specific molecules that you want. So it's kind of the same deal. And I do have a diagram of that as well to show that after this. But similar kind of principles work. It has a permeate. It has stuff that's retained, which they actually call the retained stuff in reverse osmosis the concentrate, because it's actually concentrating all the stuff you don't want. And then the permeate includes all of the nice drinking water that you're eventually going to want. So here's my thing talking about dialysis again. So I kind of just drew a diagram. But you can see up here, you'll just have basically a pump that's pumping your diafiltrate or the solution that you do want. And then you have all of the stuff that contains you have all of your product or whatever it is that you actually do want to keep. Why is the marker not drawing? It's just the wrong color. Yeah. You have all the stuff that you do want to keep is in here. But also you have the buffer that you don't want to keep, like the urea. And so you just basically go through the whole process, and out comes all the stuff you don't want, and you're replacing it with the stuff you do want over time. And then there's reverse osmosis. These are like reverse osmosis membranes. Again, they have the same kind of thing, although they tend to be long cylinders. Just gives it an extra surface area and the extra way that you wrap the membranes around and everything. So It's the same kind of principle. You got your feed to come in, and then out you'll get your permeate, and out you'll get your concentrate, as they call it in water treatment, but the retentate, essentially. But I went for it. There we go. And that's what an industrial scale reverse osmosis unit looks like. Lots of water plants will have stuff that look like this or even bigger. Because they're just doing it on such a large scale, right? Back a couple images ago, I showed you like this guy. No. I showed you that guy, but let me get back to the picture. I showed you this guy, right? Proportionally to my avatar size, it's about this tall and about this thick. I work with one of these all the time. And so this thing is tiny. We're doing like maybe like 2 or 3 liters of solution through something that's this small, right? The next size up is basically this guy. You can stack multiple cassettes in it and run it parallel. So you can have multiple filters in it, all running parallel with each other. And that one we use for like 200, maybe 300 liters worth of stuff. But, you know, these guys are doing millions of gallons a day, potentially. You'll have a bunch more units of these, but like these guys see some serious see some serious work and see some continuous movement. But that's all the basics of the whole process, and I can get a little bit more detailed. Boomin'? Thank you. I don't know why I read it.
30 minutes.
Oh, 30 minutes. Alright, but yeah, that's the basics I wanted to talk about. So I know B-Tech might have wanted to talk a little bit more about chromatography machines and stuff, but other than that, no.
Minutes, so that was actually 30 minutes exact, so good job, actually. Chemists secretly have very good timers in their heads. So, I myself, I had a bit of a plan on following this up a bit because, as you heard, that I also do chromatography as my job. Normally, I let the lecturer do the whole thing, but this was the whole 30 minutes. I think we're actually good, and this is a very good primer to anything that I talk about. What I would talk about is the kind of pre-baked solutions that's used in commercial chemistry, and it would be less about the theory and more about the application and what this is used for. And in a broad way, I love to just add a one point, like that chromatography is one of our tools as humans to reverse entropy, which is something that plagues every aspect of our lives, and this is actually something that directly separates and orders things for us, which is extremely useful. I know that it kind of comes off as very abstract and weird sort of idea, but this is something that can be used in many different parts of your life. I guess a very basic example could be distillation. You know, you're separating one from the other. Although chromatography has a different root. You know, in fact, it came from the Greek word chroma, color, photography, to separate. So it's separating colors, you know? Anyways, so thank you very much, Enbien, for the lecture. This was wonderful. Enbien, again, is our moderator, and if he's been part of the group for quite a while, and I wanted him to talk about his work for a while. I actually want a lot of you guys to talk about your work or really any special interests you guys may have. At this point, I have half a mind to do one on modern sports sword fighting because that's a huge part of my life, and I do want to have special interest lectures too. We are a STEM group, by the way, for anybody who came in and didn't hear. I've been doing this so many times, I forgot to do the most basic thing, you know? Hi, this is DegenTech. We're a STEM professional working group. For anybody who walked in previously and had no idea what was going on, we have a monthly meetup. This is the lecture portion of it. We try to talk to each other, get to know each other a little bit, and we get to know each other so we have the resources of the different specialists that you meet in VR, just hanging out. Like, we do this just, you know, because we want to. It's just fun for us. And, you know, some of us, I myself don't like talking about work, but I love talking shop, you know. I love having those technical talks, and you guys definitely scratch that brain itch in me. And some of you guys are also scary smart a little bit. But we also do try to have broad, all levels, all types of skill sets, so please don't don't feel like you have to be a certain somebody. We do have the working STEM kind of slant to it. We do try to keep focused, but please, we take all types of people here. So thank you very much for this meetup. You guys make these meetups and make the group what it is. All right. So I'm going to recap with a couple of the key announcements. We have a Twitter. We have some new content on it. I'm actually, as the outro, I'll be playing our first uploaded video. And if you guys were around, it was the Chernobyl reactor walkthrough. All right. Again, also, I do sometimes hate to beg, but we are looking for some Discord Nitro boosts. We're looking for 2 of them if possible. We're just trying to spread, because it's like $70 a month basically. So if spread it around, it's a little, but of course, completely charity. So thank you, everyone. I'm going to be hanging around for a while. It's also Saturday, so fun stuff is always happening. We'll see if we can transition into something else after this. But I'm going to put the outro, which is going to be the lecture we had a couple of months ago. Our next lecture, I'm going to be honest, there might be a little bit of delay. I have some things coming up at the end of the month. So I will also be asking if anybody is willing to host a lecture or to do any sort of other activities. We're always trying to expand what we do here at DegenTech. All right, so please come talk to me, Apotech, Enbien, or I don't know, just shout at us in the Discord. We'll get the message eventually. All right, y'all. So please don't think of this as the end. It's just this is the end of the structured section. So please talk to each other. It's what the group is about, anyways. We're here to hang out. It is VRchat. All right, so I'm gonna get the video going. I'm gonna get a drink, and then I'll be out there talking with y'all. Thanks again.